Hi, everybody, and thank you for joining on time. We are going to give folks a few minutes to join, and then we will get started. Okay, when you're ready.
Hi, everyone. Welcome to the Certified Group Training on Beauty Inside and Out, your guide to allergen testing. My name is Kike Bello. I'm the marketing manager with Certified Laboratories for the cosmetics, OTC, and supplements vertical. Today, presenters team includes Dr. Jay Alapet, Chief Science Officer for Chemistry with Certified Group, John Cervello, Molecular Supervisor for Certified Laboratories, and Dr. Jay Sharoff, Senior Director for Labeling, Cannabis, and Claim Services with the EAS Consulting Group. A few notes we get started. Today's webinar will be recorded and made available on demand. All registrants will receive access to the recording when available. And if you have any questions, please enter them into the Zoom Q&A, and we will do our best to address them as time allows following the presentation. If you're unable to answer the questions, our team will follow up via email. And thank you for joining us. And I'll hand it over to our first presenter, Dr. Jay, to kick us off.
Thank you, Kiki. Thank you, Jess. Welcome, everybody. Today, we are going to dive deep into the intricacies of allergen testing. And our conversation today will span across three interconnected industries, food and beverage, cosmetics, and dietary supplements. Why? While these industries are regulated differently and how the customers use these products, there is a common threat or a challenge in these products, allergens, which is undeclared sometimes, then unexpected, and often misunderstood. That's the reason we are bringing these three segments together. You will notice today's presentation, myself as well as my co-presenters, John and Majid, will pivot between the food and beverage, cosmetics, and dietary supplements. This is by design. The reason is whether it is a peanut allergy in a snack bar, or a milk residue in a probiotic capsule, or a fragrant molecule in a cosmetics, the underlying science behind testing, the stakes for safety, and the regulatory pressures are equally critical for all these sectors. That's the reason you'll see we pivot between these three segments. Now, what you learn from one segment can be equally applied to the other segments, what we are going to discuss, because as of now, as we are all aware, the shared supply chain, the clean label demands, and global regulation are extremely important for these three segments. So let us get started. So the plan today is to give a brief introduction of who we are in the certified group and how can we support you in your food safety programs. So we'll introduce a little bit of the allergens in food, cosmetic, and supplements, then navigating a little bit on various regulations. Then other important question always asked is, what is driving this demand for testing allergens? Then we'll get into the details of the testing technologies. We talk about ELISA testing, which is a gold standard. We talk about the PCR, and specifically for chemical testing for cosmetics allergens. Then what are the challenges in allergen testing? We're going to get into what is a false positive, false negative, what are the inferences, and how we try to fix those challenges in our laboratory. Then, as I mentioned, how certified group can help in your allergen labeling and detection techniques. And Majid will come back with the allergen labeling essentials and what are the key takeaways from this webinar. Now, as I mentioned to you, we recognize these three segments, the food, the beverage, and the cosmetic and dietary supplements, are equally important for the allergen testing. That is what you see our expertise is, the certified laboratory, cosmetics, OTC, and supplements. We have food and beverage. We have the consulting services. We have the regulatory services. So as all these groups together as an organization, we do things so the world can trust in what it consumes. So these are the various parts of the certified group. Now, we clearly recognize the importance of proximity as well as the responsiveness. Especially in the allergen testing, is important how soon your sample can get into the lab, how soon we can respond to your demands or your request. It is extremely important. So you can see our footprint all the way from Mexico and most of the parts in the United States to Canada to Europe. So we are present everywhere. So we are never far from the fast, reliable, and regulatory compliant testing for you. This slide shows what our general capabilities. Definitely, allergen by ELISA and PCR is our topic today. We can test all the major big allergens, numerous ELISA platforms, automated testing services, fast turnaround time. We'll dig deeper into those testing in detail. Now, what else we can do? We can do the proximate analysis, minerals, vitamins, amino acids, including the PDCAS, how good the qualified your protein is. And dietary fiber, all the way to low molecular weight soluble dietary fiber. Then radicity analysis, which shows the quality of your product, TBA, anicidine value, peroxide value, filth and decomposition. This is also important these days. Then remember, all these are done in a green, sustainable way. That is our vision as we move forward in the new times. Then accurate precise solutions for pesticide residues, heavy metals, antibiotic residues, beta agonists, mycotoxin, artificial colors. So we can do most of the residues and contaminants in various of our laboratories. Then we are also focused on emerging risks like PFAS, phthalates, iodide, and iodide. These are becoming emerging risks for the industry. We can do those kind of analysis. Now getting to our main topic today, the allergens. What is food and dietary supplement allergen? It's an allergenic reaction that can happen fast, within minutes of the consumption of or exposure. And it is a specific immune response that occurs with an exposure to a given food. This is, I'm talking about dietary and supplement allergens. And this is different from food intolerance. People often confused with what is an allergen, what is an intolerance. Now, intolerance is an adverse reaction to foods, but that does not involve immune system. Now, how do we get into this problem? Entering the food supply by breakdown in processing. There may be labeling challenges, so people do not know what they are eating or drinking, and storage as well as communication gaps between the stakeholders. What is the other reasons this is becoming more and more problem? The climate change, the changes in the crop genetics, and environmental stresses also influence the expression of genes and the new proteins or allergens are being added into the food supply chain. Then undeclared allergens responsible for a high percentage of food recalls that you'll see in the next few slides. One of the key takeaway message from this food allergy here is 33 million people, it's a pretty significant amount of people suffer from food allergies. And 1 in 10 adults and 1 in 13 children. That is significant. And we also call it as it's a food allergy epidemic. Why? Because there are 377% increase from 2007 to 2016 in anaphylactic extreme cases of allergen reactions called anaphylactic reaction. We'll talk about that a little bit later. So that is the reason which is called epidemic. And again, once in every 3 minutes, somebody get into an intensive care unit for anything related to allergens. Then 50% of the incidents has become very severe in the case of adults and 42% become severe in the case of children. That is the challenge we are facing these days with food allergy. This again show what is the reasons for food recalls in 2024. You can see undeclared allergens correspond to 34% of food allergen recalls. Combined together with the salmonella and the listeria, if you these put together, salmonella and listeria, the food allergy recalls are higher than these percentages. There are other reasons the food are recalled. Now we get into the cosmetic recalls. So you can see that the major cosmetic recall problem is microbial contamination, that is the 76%. Then we have inorganic contamination with the metals and heavy metals and so on. So these two segments, 6.6 and 4.8%, incorrect and undisclosed ingredients, labeling and packaging. And we do not have a specific data on what is because of allergens, but we assume this is going to be the place where most of the allergen-related food recalls come from. Now, so the food recalls in 2011 to 20, the cosmetic recalls from 2011 to 23, 334, affecting 77 million units of the the cosmetic recalls. Then what you'll also see, the new regulation, the MOCRA 2022, is expected to increase allergen disclosure because one of the conditions of the MOCRA Act is the adverse effect has to be labeled and reported. So that means there may be more and more incidents of allergy-related issues in the cosmetics. Now, one of the recent recall is because of the butyl phenyl methylpropanol, cited as a culprit in recent recall batches in Portugal. So basically, what the point here is there is allergen recalls in cosmetics, and they're going to increase with the new regulations coming. Now, what are the reasons for recalls in dietary supplements? The main reason the dietary supplement is adulteration and undeclared or prohibited substances, which you see what is going on, the Amazon-related testings and so on. The other one is the mislabeling and inaccurate claims. Then undeclared allergens is again a big part. The hidden allergens commonly noted in foods, but also in the dietary supplements. Then contamination and impurity issues, and safety and adverse events not foreseen. So when the product is launched, these people were not studied or not reported, but it is coming as a reason for the recalls, and issues related to the GMP manufacturing failures. So this slide show what is the significance of allergen testing. You can see in 2022 the recalls were 47%. Now that goes from 2023 it goes to 63%. So we can see the recalls due to the allergens are increasing significantly coming days. So testing them accurately is also extremely important. Now, so the food, beverages, and dietary supplements, they share a lot of commonalities, but dietary supplement has some unique issues. The botanicals or natural products which are incorporated into the dietary supplements may contain unexpected allergy proteins like plant latex. And there are issues with the labeling inconsistencies. Then cross-contamination between the facilities are using multiple products or producing multiple products using the similar units that is causing cross-contamination. Now, FALPA, which is the Food Allergen Labeling and Consumer Protection Act, clearly says the allergens has to be labeled either in the ingredient or may contain a pot. So these are some of the additional challenges for dietary supplements. So this is a comparison of the comparison between food dietary supplement allergies. For an example, both the definition by definition, both are immune reaction to food protein and the immune reaction to ingredient in a dietary supplement. And the exposure, there are the 9 major allergens. This is the same. The common allergens, the 9 allergens are the same for both food and beverage as well as for the dietary supplements. Then regulatory oversight. In the case of food, it is by FDA under FALC, CPA, and USDA. Here, FDA under DSHA and FALPA are the regulatory oversights. And regulatory requirements are the same. If it is there, you have to label it. And complexity of ingredients, this is slightly different. The food, the number of ingredients, what you're eating may be less, but due to the normal processing technologies, incorporation of novel foods and so on, that's also increasing. By default, the dietary supplement contain multi-ingredients and poorly characterized sometimes. And the risk of hidden allergens, it is lower because we know what we are eating. The dietary supplements, there are so many ingredients coming from different parts and put together in the matrix. So examples are the same. Peanut allergy is a big issues happening these days. Then the milk allergen is a big problem in dietary supplements and so on. Now, remember, both food and supplement allergies are typically type 1 hypersensitivity. It is antibody-mediated, IgE-mediated reaction, immune reaction. It can range from mild to severe situations. And initially, our system is being sensitized. Then it is being elicited in the re-exposure. That is how both the food and beverage as well as the dietary supplement allergen sequence of events. Now, introductions to cosmetics. Why cosmetics allergen is becoming important? It's valued at $354 billion, the industry is, with the strong growth in skincare, hair care, and natural organic products, and the rising demand for safe, sustainable formulation. These are all causing an increase in the testing of cosmetic allergens. On an average, 6 to 12 cosmetics are daily used by people, 6 to 12 cosmetic products. Then, an estimated 10 to 15% of consumers report skin sensitivity or allergenic reaction to cosmetic products. That also increases the incidence of testing and the request for testing. Now, regulatory requirements. FDA, the cosmetic as cosmetic could not be adulterated or misbranded, and has to be clearly spelled out and written on the labels. And the new MOCRA Act reinforces that idea. Then EU cosmetic regulation EZ 1223-209 requires labeling of 26 specific fragrant allergens, which we do not have in the United States, but EU requires if it is more than 0.001% on leave-on products and 0.01% on wash-off products. Then, importance of allergen testing in cosmetic. It is the best way we can ensures consumer the safety and protects the brand reputation and prevents costly recalls. The recalls are extremely costly for any producers. We continue with the allergens in cosmetics. The ingredients in a cosmetic product that can cause adverse immune reaction when a person's immune system overreacts after exposure. Now, we should also be able to differentiate what is the irritation versus allergy, which you'll see in the bottom. These reactions are usually classified as type 4. As I mentioned, the allergens in food and beverages and dietary supplement is type 1. Here it is type 4, often manifesting in allergenic contact dermatitis, AZD. And it can be all the way from redness, rash, itching, burning, swelling, hives, flaking or feeling, in severe cases, blistering. How does the type 4 hypersensitivity works? It's a T cell mediated. What happens in the sensitization phase, the immune system is primed by sensitizing ingredient. It could be a simple cosmetic ingredient like a small molecule, and that is called the elicitation phase. On re-exposure, immune system reacts, causing visible skin symptoms. That is the difference between type 1 allergenic reaction, IgE mediated, which is T cell mediated type 4 allergenic reactions in cosmetics. So common allergenic ingredient, as I mentioned, there are so many things being added into the cosmetics. Like an example, preservatives. There's so many preservatives are added. MIT is an example, methyl isothiazolinone, CMIT, parabens, formaldehyde releases. These are added to prevent microbial growth in the product, but they can be allergenic. Then fragrances, linalool, limonene, eugenine, cinnamon. These are the molecules added into the products. In most of the cases, we put it as a fragrant molecules. We did not individually mention these molecules. And the dyes are added, PPD, and certain things are added. Coal-tar dyes are added, which can also elicit allergenic reaction. And sunscreens, botanicals, which, as I mentioned, botanicals are coming from various parts of the supply chain, and that can also elicit allergenic reactions. Metals like nickel, rubber and resin components, latex, colophonium, thiurins, and lanolin and derivatives, they can also lead into allergenic reactions in cosmetics. Now, let us get into a little bit into the regulatory landscape of allergens. So, as I mentioned, the regulations are different for food and beverage and the dietary supplements and the cosmetics. Let us see what is going on in the food area. The EU regulation 1169 2011 is called the Food Information to Consumers Regulation, which says a comprehensive rules on food labeling in the European Union. All the 14 allergens, there are 14 allergens are to be labeled on a food label in the Europe. Ensure consumers receive clear and accurate information about food products they purchase, and so that they can make the informed decisions. So here are the 14 allergens. The difference between the 9 allergens in the US versus the 14 allergens is celery, lupin, sulfites, mollusks, and mustard. So these are the 5 molecules or 5 proteins differently or added into the European Union. Now, in the case of United States, FALCA is the one required food allergen labeling, requires that food containing any of the major allergens, 9 major allergens, be clearly labeled. USDA ensure that meat, poultry, and egg products complied with the allergen labeling requirements. There is a slight difference between the US list versus Canadian list. There are 2 additional allergens, which are sulfites and mustard added. And also, coconut is being considered as a tree nut still in Canada. So then we'll continue with the allergens in food. FDA published the key final guidance in January this year. What it says, provide a detailed labeling guidelines, including sesame, highly refined oils, flavoring, and bulk products. Then this is a question people always ask. If the product is extremely purified and if there is no protein inside that, do I have to test allergen? So this is one of the guidelines from FDA clarifies that protein-free derivatives of major allergens, example certain oils and flavors, do not require allergen labeling. That is the question most of the people, the customers ask. Then highly refined oils without protein may be exempt. The lactose or ghee still require labeling due to their residual protein. Evaluation of public health importance of food allergens other than the major food allergens, that's also required by FDA. Now, updates to FDA allergen definition, as briefly alluded to, refined tree nut list. FDA narrowed the tree nut allergens that require labeling. Coconut, along with cola, beech nut, ginkgo, chestnut, shea, they are removed from the tree nut list. Now, egg now include egg from ducks, geese, quail, and other birds. So the manufacturer has to clearly label duck egg. Then milk cows, milk from other domestic ruminants like goats and sheep. So as I mentioned, goat milk has to be specifically labeled. Another important thing happened in the regulatory area is California Senate Bill 68, which requires Senate Bill proposes requiring restaurants, including school cafeterias, to disclose major allergens, including sesame, on the menus starting July 1, 2026. It passed, currently navigating through legislative committees. Now, what is happening in the regulatory landscape of dietary supplements? It is, as I mentioned before, regulated under DSHA. FDA is CFR Part 111 for GMPs. No mandatory allergen labeling like FALPA, but if allergens are present, must be declared on the supplement facts, either in the supplement facts or may contain part. High risk cross-contamination during blending and encapsulation. FDA recognized that. It is important that if your product is blended in different locations, brought together for formulations, so you should clearly look into the ingredient list, study it, and the safety part, and try to understand the allergens present in it. Testing is not mandated, but increasingly expected to support label claims and consumer safety. Now, what is the regulatory landscape of allergens in cosmetics? So the main one is EU regulation 1223-209 and US FDA guidelines. What all these say? Ensure safety for human health. That is what the main theme of all these regulations, and declare allergic fragrance substances. So European Union has specifically said the 26 molecules, if it is more than 0.001% for leave-on products and 0.01% for wash-off products, has to be clearly labeled. Be tested for dermal sensitization in pre-market safety assessment. This is always the question we ask. If my product has to be hypoallergenic, what are the things to be done? then regulator under Federal Food, Drug, and Cosmetic Acts in the United States. And of course, the new law, it is called the MOCRA, which is passed in 2022. It is going to change the landscape of allergen labeling in the United States for cosmetics. Then allergen disclosures, not required under FALPA, but EU requires 26 cosmetic allergens to be listed if above the threshold, as I mentioned, 0.001% and 0.1% for 0.1% for the wash-off products. Then US FDA focus on safety substantiation. That is exactly what the MOCRA is going to do. If there is an incidence of reported side effects, it has to be reported immediately. Then, what are the testing to be used? The analytical testing, the ELISA, PCR, LCMS are increasingly used for claims validation, whether it is for hypoallergenic claims or for free from particular allergens claims. That is where the regulatory landscape related to food and beverage, dietary supplements, and cosmetics are now. Now, what is, as I mentioned, what is driving this allergen testing? So when you look holistically, there are three parts. One is the business impacts. Allergen recalls are expensive. Number two is the consumer trends. So people are looking for free from products, free from allergen products. And of course, there is international trade pressures. As I mentioned, 9 major allergens in the United States, 11 allergens in Canada, 14 allergens in Europe, and Asian regions, they have a different allergen list. So these are all the reasons that is making the customers, okay, let us do all the allergen testing upfront before they import or export the product to particular locations. Here you see a case example, FDA 2024 data. 48% of the US class 1 recalls, as I mentioned, were due to undeclared allergens. That affects the bottom line of the business, higher than pathogens, and median direct cost per incident for a CGP is $82 million. So that is one of the reasons people are focused on validated and verified allergen testing. So what is driving the allergen testing and dietary supplements? Of course, the regulatory compliance, FDA, Food and Allergen Labeling, Consumer Protection Act, FALPA, and the Food Safety Modernization Act requirements for the allergen controls. Then consumer safety. That is another big reason. The brand protection, global supply chain, as I mentioned, the supply chain is so global these days, so products are coming from everywhere. It is important what is where the products are coming from, what are the potential allergens, and complex matrices. The complex matrices also not only the inherent allergens, the processing can change the structure of the proteins, the epitope of the protein, and some of the proteins can become allergenic. Then market differentiation, people always want to differentiate what their product, how the product is different from the competition, industry growth. So what is causing the allergen testing increase in the allergen testing in cosmetics? As we see it here, the regulatory pressure, the EU cosmetic regulation, the FDA oversight, as I said, the MOCRA, the new regulation, the ISO standards, they are all the reasons for increasing the allergen testing in the cosmetics. And of course, the consumer demand for free from hypoallergenic transparency expectations, they are all leading into the increase in the allergen testing. Of course, the brand protection, global supply chain, innovation and differentiation, how your product is different from the rest of the products in the market, and allergen tested claims gives a premium price value for the products. Then food to cosmetics spillover. We have learned a lot of things from the food and beverage and dietary supplements that is crossing over to the cosmetic side because of the introduction of natural products, the sustainability trends, natural extracts and popular, but they can also lead to allergenic reactions. Then testing ensures safer use. So these are the trends or the reasons where you see an increase in the allergen testing across various segments. Now let us move into the technologies. The common food allergen testing technologies. So as I mentioned briefly in the beginning, detection of proteins, that is still the gold standard for allergen testing. It's called enzyme-linked immunosorbent assay, ELISA. This is the gold standard. We'll talk a little bit of the science behind it. What are the challenges of the ELISA testing in the coming slides? The detection of DNA polymerase chain reaction based methods. John is going to talk about the details. Again, the sensitivity is extremely high, and there are places where you can use PCR much better than ELISA. Then detection of proteins and peptides using LC-HMS. This is still in the infancy. People are still figuring out what is the best way to make it accessible and cheaper for the customers. That's going on. Then, as far as the chemistry testing are concerned, especially for the cosmetics, there are HPLC, GC, HPTLC, HPLC-MS, GC-MS, etc., with various detection systems, as diode array detection, FLD, fluorescent detection, and so on. Let us get into the details on so talk about the ELISA system. As I mentioned, in ELISA, we are testing what you call the proteins. The proteins are the one, or the antigens are the one which is causing the trouble, the allergenic reaction. So without knowing the protein, if you start testing the product, it is extremely can create complicated results. So knowing the protein is important for ELISA testing. In the case of soy, it is glycinin and corn glycinin. In the case of egg, it is ovalbumin, ovimicoid, and so on. And milk, alpha, beta, casein, and beta lactoglobulin, they can cause allergenic reactions. The peanut, ARA-H1, ARA-H2. Then almond. So these are the various proteins causing. So protein is one, and the protein which is causing the allergenic reactions are extremely important. Knowing that one is important for any kit manufacturers, because based on that protein, its structure, its epitope, that's how they develop the test. How ELISA works? So ELISA, there are 4 different kinds of ELISA, enzyme-linked immunosorbent. One is direct ELISA. So we have an antigen here. Then you're adding the antibody. So antibody will have an enzyme linked to it. Then later you add a substrate. The substrate will change its color. The enzyme change the color of the substrate, and that will respond to the amount of protein present or antigen presence. So you have the antigen, the antibody, the substrate, and changes its color. And the change in the color is the reason for the increase in color. So we can quantify the amount of allergen present. Then there is the indirect ELISA. Again, the theory is the same. Here you have the antigen. They have a primary antibody hooked onto the antigen. There is a secondary antibody with the enzyme, and this enzyme changes the color of the substrate, then develop the color. Again, you can plot a concentration against the color, then you can find out, quantify how much allergen is present. The sandwich ELISA, which is the most common ELISA which is deployed in our laboratories, is slightly different. So we have a primary antibody. So it's called the capture antibody. Then you have an antigen. It is sandwiched between a secondary antibody. It's called detection antibody. The rest is the same. You have an enzyme linked to the secondary antibody. Then the substrate changes its color by the action of the enzymes. Again, the color developed with the concentration. By plotting the graph, we can find out the amount of allergen present. Competitive ELISA is slightly different, where the antigen is mixed with the antibody, and the remaining antibody is docked onto the antigen, and you can find out the color. So the response of the color will be inversely proportional. If you have a higher concentration, you have a lower response. If you have a lower antigen, you have a higher response. That's a slight difference. So 4 different kinds of ELISA testing. So what is the advantage and disadvantage of ELISA? So as I mentioned, use antibodies to selectively target the antigen. So there are monoclonal or polyclonal antibodies. So when you go for a monoclonal, it will be very specific. So the problem for false positive, false negative will be very low for the monoclonal antibodies, but at the same time, the cost will be very higher. So as I said, the advantage is highly sensitive and specific, is cost effective, easy to use, high throughput, and we use that in several of our laboratories across the country. Then disadvantage is cross-reactivity, which we'll talk about a little bit later. Then limited detection, and it is time consuming sometimes. Then often leads to false positive and false negative. So this is how, as I mentioned, when you add, you have the antibody here, we have the antigen, we have another antibody. Then what happens is the color development with concentration. You can see based on the curve, we can figure out, okay, what is the concentration of the antigen or allergenic protein. So we have to do the proficiency test to make sure our testing is well performed and verified. So we also do positive and negative controls. We do calibration controls. So we do the extraction environment controls. What I'm saying is these controls are being used to make sure the data we produce in the lab are reproducible, repeatable, and reliable. So now we move into PCR. John is going to take on another important technique what we have in-house to support our customers. John?
All right. Thank you, Jay. So my name is John Cervello. I am the molecular supervisor at Certified Labs. I will be going over a bit of the science behind PCR and its applications to allergen detection. First, what is PCR? So PCR is a molecular biology technique to mass produce copies of DNA through a process called DNA amplification. You can think of it as a molecular photocopier where we start off with a tiny amount of DNA and make millions of copies in such a way that we can detect it with an instrument. So the most important step before we get anywhere near that instrument is sample preparation. Each type of sample requires specific consideration for DNA extraction. Here I have outlined some of the problems we might come across with each matrix. For food, we need to consider processing effects as well as the ingredients. That would be things such as heat, acid, or anything else that might actually either denature the DNA or just make it hard to detect. For supplements, we have excipients and coatings, as well as the fact that sometimes there is no viable DNA left after production. Sometimes these, for example, oils or sugars or things like that might just be so purified that there shouldn't be any DNA left after the final product. For cosmetics, oils, pigments, surfactants, and preservatives are the main concern. What these all do is either interfere with the DNA extraction or provide inhibitory effects on the PCR process. Either of these could lead to false negatives if they're not addressed. The key item that I want to focus on here is fats or oil interference, as this is the most relevant to supplements and cosmetics. What happens is that if sample contains lipids, emulsifiers, or detergents, these can form complexes with the DNA that will trap and prevent it from binding during extraction. To address this, we use hexane in a process called defatting to dissolve and break apart these complexes, allowing the DNA and proteins to separate into a water phase. This water layer can then be used in DNA extraction. The extraction process itself is actually quite straightforward. Each cell contains an outer membrane and a nucleus packed with DNA. We use a special kit designed to break apart the cells and separate and purify the DNA from all other cellular materials and proteins. The end result is really just a little tiny vial that should only contain water and DNA. After extraction, we combine the contents of this little vial with all the parts needed to mass produce the DNA. We use kits that contain highly specific primers that target and bind special regions of the DNA unique to the species that we're looking for. Now, this could be plant, animal, or whatever might be the case. From this primer binding site, the polymerase attaches and makes copies of each DNA strand. Through successive steps, more and more copies are created. Here on the little image I have, denaturation is DNA strand separation by heating. This is one of the key points here where we're actually separating the strands. Annealing is a step where the primers bind to specific sites on reciprocal strands. Extension is a process where the polymerase combines the ingredients that we have included into this little vial to replicate each strand, and the cycle will continue. How this works on the instrument is through heating and cooling cycles that allow the DNA to go through a process of strand separation and duplication. With each cycle, the amount of target DNA doubles, starting with a tiny amount of DNA and exponentially growing to potentially millions of copies. The instrument then uses fluorescence to measure in real time the DNA produced in the reaction. So in each one of these little cycles, we'll be able to tell how the growth continues. And that gets a little clearer on this next slide. So here we have a sample of the data produced by the instrument in what we call the exponential phase, which is that very first section before the dotted line. The target DNA is doubled every cycle. It will eventually reach a plateau phase when the ingredients to make more DNA run out. The more target DNA present in the sample, the sooner this will occur. So you see all the lines that are further to the left, when they increase, those chances are those had a lot more of the target DNA than the ones later on. In the first part of the exponential phase, there's not enough DNA to be detected, which is why you see kind of that flat section that I have in that little box. But eventually, as DNA is replicated, it will begin to be picked up by the instrument. And this is picked up by fluorescence due to part of the reaction of the DNA duplication. For reference, the green line along the bottom shows a negative result with no noticeable amplification. That usually either means that the reaction failed or there just wasn't enough DNA in the sample to begin with. Our limits of detection are actually quite low. Sometimes they're 1 ppm or below. Okay, so in this section, I'm going to go over a bit of the pros and cons. Pros, the PCR will detect even trace amounts of DNA, which is useful in possible contamination or when you're dealing with residues. This can be an issue whenever you're changing between different products on a facility and you want to make sure that nothing was left over after the clean. With some kits, we're able to multiplex and test several allergens at once. What that means is that the costs can really be brought down, which is very beneficial considering, that one of the cons later on. Lastly, PCR is more resistant to matrix interference. So sometimes when we deal with some of the other methods, just the fact that there is either acids can denature proteins or heat can cause problems. So DNA is a little bit more stable and we're able to work with it as an alternative sometimes. The downside is that DNA itself is not an allergen. We're just using DNA as a proxy for possible other allergens present in the mix because it's a little bit more stable. Allergens tend to be proteins and in some cases chemicals. These would not be detected by PCR. PCR also tends to be more expensive due to kit costs and really specialized equipment. Last, PCR is not suitable for egg or milk allergens, which I could definitely go into more detail if there's questions. So whenever you're trying to make a decision of which test to pick, there's a few things to consider. Again, PCR detects DNA as opposed to actual allergens. It's typically qualitative instead of quantitative. DNA often does survive heat, pressure, and pH changes, which makes it ideal in certain situations. And DNA can be detected in samples where there is no functioning allergen proteins left in a sample, which is actually a bad thing because this would mean that you have a false positive. There might be DNA left where all the proteins were denatured by the heating process and are no longer going to affect or elicit rather an allergic response. Lastly, PCR is higher on that cost spectrum. Ultimately, it may be necessary to perform a validation if the type of test to choose is not really certain. My last slide here are the kits that I would be using and their associated limit of detection, which is around, well, I think it's 0.4 ppm. And at this point, I'll turn it back over to Jay.
Thanks, John. Thank you very much. So we have seen the two important or to a certain level complementary techniques. So whenever ELISA fails, our go-to testing is PCR. So another technique which is being still, I would say, is infancy is the LCMS method, where ELISA fails or we can't get a lot of information from ELISA or PCR, we go to LCMS. The problem with that one is it is very expensive and we need a lot of technical expertise who can analyze the data and time consuming and maintenance. So here what happens is allergens, like John mentioned, allergen extracted with appropriate solvents, extracted protein is digested with the enzymes like trypsin, and we need to have an additional step of protein cleanup, which we don't require in ELISA, then HPLC-MS detection, and of course the data analysis. So it is a little involved, complicated, and expensive procedure. Now, in the case of, as I mentioned, the food and dietary supplements allergens is type 1 hypersensitivity. This is cosmetics is type 4 hypersensitivity is the T cell mediated because the culprits are mostly smaller molecules. So what are the techniques we can use for the testing those allergens? Fragrant allergens, for example, 26 molecules listed by EU. We use GCMS, it's a gold standard for volatile, semi-volatile fragrant molecules. And of course, this involves the sample preparation and injection either as a solution or the headspace SPME techniques we can use. Then provides the quantitative profiles against regulatory thresholds. As I mentioned, 0.001% on leave-on and 0.01% for rinse off. Then GC, we can use FID techniques. This is alternative to GCMS when only quantitation, not identification is required, and useful for routine batch screening once identity is confirmed. So these are the techniques we use in our laboratories for allergen cosmetics allergens. Then preservative and allergens for a slightly bigger molecule, which has a chromophore, we can use HPLC UV, or in case of multiple chromophores, we can use diode array detection for parabens, etc. We also use LCMS for low-level detection of complex matrices. Then continue with the chemical methods for in cosmetics. As I mentioned, the metals can be allergenic. In the case of cosmetics, we use ICP-MS for ultra trace level detection, the PP level for nickel, chromium, cobalt, and so on. We also use ICP-OES and atomic AAS spectroscopy for alternative or higher detection limits for routine screening. In addition to that, there are some other techniques we use in our laboratories. FTIR, used mainly for screening polymers for residual monomers or allergenic with allergenic potential. We also use thin layer chromatography. It has not been very common these days, but it is a very easy, readily available technique for faster detection of allergenic molecules. So this shows what are the common ELISA kits available in the market. For example, for all the major 9 allergens we mentioned before, there is a commercial kit available with various limit of detection and limit of quantification. This is extremely important for the laboratory. So when we vet a kit for a technique, what we'll do is, what is the epitope they are using? Are they using a monoclonal or polyclonal or monoclonal antibodies? What are the reported in the evaluation process? What are the false positive? What are the false negatives? What are the interferences they are reported? We'll study all this before we bring that particular kit into our laboratory for testing the customer products. Now, next few slides, we are going to see what are the challenges in the testing allergens. As I mentioned before, these are all proteins. So the protein has to be extracted. So the food matrix effects are extremely important, how efficiently we can take this protein out. So food matrix can unique properties, chemical or physical, that can interfere with the detection. For example, as I mentioned, these techniques are all color reactions, spectrophotometric observations. So whenever there is a ingredient, a chromophore, which can either increase the chromophore intensity or decrease the chromophore intensity, it can lead into the problems in the quantification. Now, how easily allergens can be extracted, as I mentioned, is extremely important. Then homogeneity and dispersion, because allergens are localized in various parts. So if you have a pound of material, it is extremely important how we homogenize the product before testing the sample. Then processing effects. As I said, the proteins can change its nature, its structure, its epitope on the processing effects. So based on the processing effects, the reaction or the results what you're getting is going to change. They're also leading to false positive and false negatives. Then emergence of novel foods. As I mentioned before, we know how the existing proteins behave, but there are a lot of proteins that are coming from a fermentation, from synthetic proteins are coming, and plant-based products are coming. This can all lead into the challenges in testing the allergens. Then cross-reactivity, as I mentioned, this is a antigen-antibody reaction. Based on some of the antigens will have a structurally similar epitope, so that can give into a reaction, and we'll assume the protein that is giving is an allergen, but it may not be. So it's extremely important to vet the cross-reactivity reported in the validation document of the kit before we bring that kit into in the laboratory. For example, proteins found in cavonna seeds are found to cross-reactive to those found in mustard seed. So we are extremely careful what kind of test kit we are going to use. When choosing commercially available methods, it's important to review the kit's validation reports for potential cross-reactivities. Now, these are our capabilities across the region. So we have Aurora testing capability, allergen testing capability in Aurora, Melville, New York, San Antonio, Texas, Atlanta, Commerce testing in Burbank in California. So we have the presence, as I mentioned, our footprint is very close to where you are. So we know the importance of proximity and the responsiveness, how faster you want the research. So we have the allergen testing capabilities all across the country. Then we have ELISA and PCR platforms, then capability to test all major allergens. And we also have automatic testing capabilities. We use the two automated systems, Chemwell and Thunderbolt. Then we have the ability to test raw materials, products, swabs, and we have also capability to test all the molecules we talked about in the cosmetic, the 26 plus. It is getting to 80 molecules in Europe now. So we have the ability to test much more than those listed allergens. So again, one of the most important technique we use for all these cosmetic molecules is GCMS. So all these molecules plus more we can do. We also go for, if it is a lower level, we go to HPLC-MS, HPLC-MS, and so on. Now we'll move into about the labeling. So we have our expert in expert services, Dr. Majid Sharaf. He's going to walk you through what we need to know you need to know about labeling of allergens. Dr. Majid?
Thanks, Jay. Hello, everyone. My name is Majid Sharaf. I'm a senior director at EAS Consulting Group. My team is responsible for labeling support, safety, and safe substantiation. In addition, also to claim substantiation and import detention. So going back to the presentation, the Food Allergen Labeling and Consumer Protection Act of 2004 requires that food and dietary supplement labels must identify the food source of all major allergens. Those 9 major allergens were mentioned by Jay early in the presentation, so I'm not getting into that. And this requirement is met if the ingredient, the common or usual name of the ingredient, contains the name of the food allergen. For example, if you have one of the ingredients is buttermilk, because the name contains the word milk, so this requirement is already met. In addition, also the regulation requires that all nuts are listed by species, same as all fish included in a product must be listed by the species name, and also all crustacean shellfish must also be listed by the species name. The food source of all major allergens must be identified on the label in at least one of 2 places, either in the ingredient statement, where the major allergen is listed as one of the ingredients, for example, milk. You can have milk as one of the ingredients, or if the ingredient is produced or is sourced or is produced from one of the major allergen, then the ingredient is listed, followed by the major allergen, also listed in the ingredient statement. For example, here we have casein. Casein is coming from milk, so you put casein, and then you follow by the word milk. Similarly, also you put tahini, then followed by the word sesame. We recommend that for when you have hydrolyzed protein, in addition of listing soy and wheat, we also recommend that if the hydrolyzed protein is coming from another source, such as meat, it's also, we prefer that you list that source so that it doesn't, the information is clear and there is no confusion. Similarly, if you have gelatin as one of the ingredients, also it's better, fish is the requirement, but also we recommend that you list the source if it's not fish, so that the information is clear and there is no confusion. Next slide, please, Jay. So these are the previous slides were for the added ingredients. So if I'm having milk in my product, then milk must be listed in one of those two places. I missed to mention that the other place is the contains statement. So in addition to the ingredient statement with the major allergen is listed, or the ingredient with the major allergen is listed, you have also another location with the right after the ingredient statement or next to it, you can add the contains statement. So for example, my product contains milk. So these are the two places where you need to include or identify the source of the major allergen in food and dietary supplement. The law requires that you identify the source in at least one of these two locations. What about allergens or coming from as an indirect addition to the product? For example, cross-contamination from manufacturing. For example, if I'm using utensil, I'm using it for an ingredient that's an allergen, then I'm using the same utensil for an ingredient that's not an allergen. This ends up in a cross-contamination. Similarly, if I'm using the cooking oil that I'm using for an allergen, then I'm using the same oil for a non-allergen. This will introduce some of this allergen into the product. In this case, for example, the cross-contamination, the presence of allergen is not defined in the law. So in this case, it's not required that the company lists the cross-contamination of this major allergen in the product, but companies select to be safe and include statements such as this product may contain one of those major allergens, or this product was made in a facility that process nuts, for example. So this way, the company selects to be on the safe side and include that, provide a cautionary statement that this product may contain one of those major allergens. With this, I return it back to Jay.
Thanks to Dr. Majid. So we'll conclude our webinar. We have seen all these various testings and what are the challenges, what are the regulations, what is driving the industry. So as I said, the food and beverage as well as dietary supplement is a little bit more evolved compared to the cosmetic allergens. So the cosmetic industry growth drives greater focus on safety, transparency, and allergen control. So allergen is cosmetic. The fragrances, the preservatives, dyes, botanicals are a significant cause of skin reactions in 10 to 15% of the consumers. Then regulations like FDA, MOCRA, EU regulations mandate labeling. So it is going to be more and more challenging moving forward. The testing methods, what we normally use, ELISA, PCR, LCMS, provide complementary tools. Choice depends on the matrix, the sensitivity needs, and the regulatory acceptance. And as I mentioned, the challenging in testing allergens is cross-reactivity, false positive, false negative, and we are in our laboratories, we'll make sure that we address these challenges before we bring a test kit to a particular product for testing. Now, certified groups role is delivering validated, defensible allergen testing that supports compliance, protects brands, and ensures consumer trust. As I mentioned, our expertise in food and beverage, cosmetics and dietary supplements. That is the reason we are able to bring forward a holistic approach in the challenges in the allergen testing. And I also mentioned we recognize the importance of proximity to your products, as well as the responsiveness. That is how we can collaborate and develop methods for your particular products. Now, I will leave to you, Kiki.
Thank you, Dr. J. Do we want to answer one or two questions that we've received? We have one question from our webinar attendees, can we completely remove allergen testing for tree nuts? In the US already removed the coconut as a tree nut allergen, if this is the only tree nut allergen we have in the facility? Dr. J?
That's an interesting question. People always ask, okay, if I use only coconut allergen, why should I test all the tree allergen? So the answer is no. You cannot completely remove tree nut allergen testing solely based on FDA's reclassification. What you need to do, if tree nut is the coconut is the only one, and if you can prove that tree nut is the only one using ingredient review, the supply controls, as well as facility mapping. Meaning that the tree nuts, no other tree nuts are used other than coconuts are used in any of the shared locations, and documentation of those in the safety plan, which if you can put together, we can avoid the labeling with the coconut.
Perfect. And then one other question we had is, as allergen thresholds are not standardized, how could a manufacturer decide on the choice of selecting a specific product testing solution? Dr. J or John? Whoever.
John?
I kind of touched a little bit on this earlier. This might be something where we have to actually go through a validation process, something called a spike validation. In short, that's something where we will get your product, and at some point, either in a finished product or as part of the manufacturing process, we might introduce a known quantity of allergen. And then once we have that product with the allergen in place, we're going to try different methods to make sure that we're able to detect it using one of the various technologies. So in the case of ELISA or PCR, just making sure that after it goes through processing and extraction, that the instrument can pick it up. So it's not always something that can be known right away. Sometimes we already have a validated method in place, but in the case we don't, we might have to go through that.
Yeah, I will also add to that some of the useful references come from White Towel, which is a voluntary incidental trace allergen labeling, which gives some kinds of controls, like for an example, peanut 0.2 to 1 ppm or gluten 20 ppm for FDA products. So basically what we need to do is what is the exposure of final products? We should understand that before doing the testing. And why you are testing? It is for regulatory compliance, it is for label verification, or market requirements.
Perfect. We have one really good question. This is for Dr. Majid. Are they a set allergen testing we need to conduct that we would cover global sales?
I'm sorry, I was in mute. What was that again?
So are there set allergen testings we need to conduct that would cover global sales?
I think I will ask John to help with this. It is a testing question. John, if you may.
I can maybe I can add a little bit. So there is no globally standardized test panel. So what basically need is a core panel, which is the 9 allergens in the United States, 11 allergens in Canada, and 14 allergens in Europe that can be tested if depending on where you are sending the your product. Then your strategy, any testing strategy should be tailored. Okay, what is that for? It is regulatory limits or it's a labeling purposes and so on, where you're sending the products. Maybe John, you want to add anything?
No, that's exactly the best method there.
Well, we are actually going to wrap it up. Thank you everyone for joining our webinar. I hope everyone has a lovely day. We are going to be sending you a link for this webinar. If you would like to please just give us your feedback, we would appreciate it. And I hope everyone has a lovely week.
Thanks, everybody.